Goodness me, technology has changed. I mean, the chemistry lab that I did my undergraduate and then doctoral research in, and the ones I did my um, postdoctoral research in, well, I suppose they'd probably gained fume cupboards since the Victorian era. But a Victorian chemist would have recognized them pretty much. It was glass apparatus in a fume cupboard. Analysis was by thin layer chromatography. We did have NMR when I started my research career in 1982. 60 megahertz NMR was what we used for routine analysis, and we had one high field NMR spectrometer, 300 megahertz, in the entire department, in what was one of the top UK chemistry departments. You're part of a team together, and drug discovery has always been and still is very much a team activity. It's not a lone genius working in an ivory tower. So the team, you need everybody's insights, and you're typically having to do something that hasn't been done before in some way or other. And so you need to come up with creative ideas, and
hello and welcome to Meet the Medicine Makers. I'm Menasha, an early career drug discovery scientist, and this podcast shares real stories from the people behind how medicines are developed, from early research through to real-world impact. Today, I'm joined by Matthew Perry, a medicinal chemist who has spent 37 years working in the field. Medicinal chemistry sits at the heart of how many medicines begin. In this episode, we'll explore Matthew's career in medicinal chemistry, how drug discovery has changed after over nearly 4 decades, and what a long career in the field can teach us about science, technology, failure, and the people behind how medicines are made. Matthew, thank you so much for joining me.
It's a pleasure.
So you've recently reflected on 37 years working in medicinal chemistry and drug discovery. Firstly, congratulations. So my first question is, how did you first find your way into chemistry, and what specifically attracted you into medicinal chemistry?
So it's one of those things I can remember as a 13-year-old thinking I want to be a chemist. So quite early in my school career, I became fascinated by chemistry, and that persisted. So I then studied science, A-levels in the UK. So I went on, and even before I got to university, I knew that my passion was in organic chemistry. So I went to university to study chemistry there, and then stayed on to do a doctorate. I worked in the group of the late Professor Sir Jack Baldwin at Oxford, and I mean, I was someone who was not really interested in biology. Biology was extremely badly taught in my school, and looking back, and so I have no formal qualifications in biology at all. But at Oxford, in Baldwin's group, there were other PhD students doing work on the biosynthesis of penicillin antibiotics, and I began to find this really interesting, and so I became more interested in the biological aspects of chemistry and the application of organic chemistry to biological problems. As an undergraduate, I'd done a course in chemical pharmacology, which I'd found extremely interesting as well. And so, I mean, at that point, for people doing research in organic chemistry, it was quite common for people to seek employment in the pharmaceutical industry. So I did a couple of postdocs, and then I was relatively young, and then applied for jobs in the pharma industry, and so that was really where I started my career.
And when you first started working, what did the early field look like?
So if you're talking about when I started my research career, I was working as a synthetic chemist, and really at that point, medicinal chemistry was not something that was done in academia, apart from a relatively small number of groups. So Professor Baldwin's group was unusual in doing what we would, I think, now call chemical biology. So using chemistry and design and synthesis of molecules, questions of biology, but that was not what I was working on. I was working on a project that was dedicated to exploring new chemistry, discovering new synthetic methods, new ways of making molecules. So the sort of general principle that I was doing was trying to develop myself as a synthetic organic chemist, give myself a really broad understanding of organic chemistry that I could then apply in an industrial career in the way that would give me the ability to make whatever molecules were needed. I mean, whether that was an industrial career or an academic career, but my aim was to explore chemistry, learn new things, discover new things.
Mm. So you started off synthetic chemistry, but then you later transitioned into medicinal chemistry. But for listeners who might not be familiar with the field, how would you explain what a medicinal chemist does?
Right. So medicinal chemistry is the science of making molecules to be tested as drugs. So as medicinal chemists, we require some kind of starting point, that is a molecule that shows activity against typically an identified biological target. And then what we do is we make a whole series of analogues and of that molecule, and we look to make changes and see what changes see what effect those changes have on the biological properties of the molecule, with the aim in the end of making a compound that is suitable to be used as a medicine to treat patients. So that involves optimization of the potency at the desired target, but that's only actually quite a small part of the role of a medicinal chemist, because a drug needs not only to be potent at the desired target, it needs to get there in the right amounts, to stay there for long enough to produce an effect, and it needs to do that without having any negative effects on the patient taking the medicine. So all of those are different aspects, a part of medicinal chemistry. So it's very challenging to get a molecule that meets all those criteria.
And what makes it so challenging?
Well, you're trying to optimize against multiple different endpoints, I suppose you can say, multiple different goals at the same time. The rules are, so every time you're guessing as to what's important, and the biological systems have a tendency to do things that you weren't anticipating, because we're using all the time relatively simplified models, at least mentally, of what we think is going on. And actually biology is far more complicated than that. You don't quite know what you're going against. So typically the target is a protein. Proteins, we have nice crystal structures of proteins, but actually proteins are not rigid. So you get a crystal structure and that's it, trapped in one form. But proteins are flexible, and proteins can adopt different conformations. which means that changes that you make don't necessarily interact with the protein in quite the same form as the one that you were thinking it had. And that's a relatively modern thing, where we even had structures of proteins. I mean, back at the start of my career, the idea of having a crystal structure of your target was kind of pie in the sky, basically. There were just very, very few protein crystal structures that had been disclosed by the late 1980s, and certainly not really of any relevant drug targets. And then in later years, most of the projects I worked on, we had multiple crystal structures of our target protein to use to guide us. But yeah, even then, it's still not an easy job.
And I mean, you've already touched on it, but how has technology in chemistry changed since you've been working?
Yeah, goodness me, technology has changed. I mean, the chemistry lab that I did my undergraduate and then doctoral research in, and the ones I did my postdoctoral research in, well, I suppose they'd probably gained fume cupboards since the Victorian era. But a Victorian chemist would have recognised them pretty much. It was glass apparatus in a fume cupboard. Analysis was by thin layer chromatography. We did have NMR when I started my research career in 1980. 60 megahertz NMR was what we used for routine analysis, and we had one high field NMR spectrometer, 300 megahertz, in the entire department, in what was one of the top UK chemistry departments. So, structural work was less advanced. But of course, we were working on the whole on simpler molecules. The arrival of new instrumentation has had huge impacts on chemistry as a science and on drug discovery as a whole. So thinking of the technologies that have come in, I mean, improved analysis methods, so far greater availability of NMR and of complex, complicated NMR analysis techniques to look at two-dimensional correlations. I mean, those have been around for a long time now, but they were pretty new. In fact, I don't remember when I got the first one of those. I don't think it was during my doctorate. But now the routine analysis by liquid chromatography, mass spectrometry, LCMS, where you can actually see whether that spot that's appeared or that peak that's appeared in your chromatogram has the mass you're expecting, or whether it's done something else. But other sort of more widely in the industry, the sort of automation techniques that came in. So when you make compounds as a medicinal chemist, those then get registered typically to the corporate compound collection if you're working in industry, and then they're dispensed to biologists for testing. Back in the old days, they were dispensed manually, using a pipette. Samples were often sort of weighed out and then even dissolved by the biologist. Now that's all automated, and the increasing level of sophistication of the automation means that smaller and smaller volumes of liquid can be dispensed, which means that you can get far more tests from a given volume of liquid, which has reduced the amount of compound that's required to be made. That automation also enabled what's known as high throughput screening, where very large numbers of compounds can be tested at one time, which is really the way that a lot of projects now start. When I started, there were a few computational chemists, but it's also been democratized so that computational chemistry, yes, there are still computational chemists, but medicinal chemists are doing things that are done by computational chemists, and that's down to improved tools being rolled out. And so when I've worked, I've always worked very closely with computational chemists, and it's kind of a very you have a constructive partnership. You bounce ideas back and forth between each other. Computational chemists are great, but they don't know that much synthesis on the whole. And you're part of a team together. And drug discovery has always been and still is very much a team activity. It's not a lone genius working in an ivory tower. It's a group of colleagues working together, bringing different ideas, different perspectives from their own backgrounds, all trying to contribute to progressing a project.
I mean, as a high throughput screener, I can fully appreciate how technology has really made my job easier. But I actually wanted to go back to something that you said. You mentioned that you used to go back and forth with your computational scientists. And I think one thing that stood out for me is that in your career reflection, you really highlighted the importance of the people you worked with. So I had a question about that. Why do people matter so much in drug discovery?
People matter because you're working as a team. You're trying to do something that's very, very hard. So the team, you need everybody's insights, and you're typically having to do something that hasn't been done before in some way or other. And so you need to come up with creative ideas, and you to really sort of think deeply, and therefore the more brain power you get, the better it is. And then often, as a medicinal chemist, you come up with an idea, but that requires that you need some kind of new assay to measure whatever it is that you're interested in. So then you have to go and talk to whichever colleagues might be able to build that assay and ask them to build it. They probably have to build a case in collaboration with you to their management as to why they should do it, because all these things cost money, they take time and effort. So that's something that person is then not doing something else in order to do that. So the people that you're working with and the working atmosphere that you have together is really important.
And I mean, can you remember a colleague or a team that has sort of changed, like in your career, changed the way you approached science, drug discovery, chemistry?
There are so many of the colleagues that I have worked with who've done things that I've thought, gosh, that's really clever. It's one of the pleasures of working in the industry is that you're working with a large number of very, very clever people. There have been people who have championed tools, technologies. So I can remember the former colleagues, the people who championed some of the sort of physicochemical understanding ideas. That was very important. The people who championed high throughput screening, the people who championed library synthesis, people whose insights into drug metabolism, into what happens, people who thought really deeply about what happens to drugs in particular circumstances. So I have worked at various points on inhaled drug projects, because my career has been partially in the respiratory disease area, and one of my colleagues who I worked with and who I discussed inhaled drugs were really deep insights that he'd gained from working on inhaled projects in the past. And the challenge essentially with an inhaled project is that you're giving it by some kind of inhalation to the lung, and you on the whole want your drug to stay in the lung long enough to have an effect, but not too long. And you don't really know where your drug is when it's in the lung or what capacity it's in, and his thinking around that was really very deep and very insightful.
Are there any other qualities that would make someone especially valuable in a good project team?
Qualities. I mean, perseverance, because you're going to fail most of the time. So you need to you need to be willing to say, oh well, that didn't work, let's try again. I think it was Churchill who said that success consists of going from failure to failure without losing enthusiasm, and that certainly applies to drug discovery. Collaborative attitude, that is essential. You need to be able to work with other people, and that relates to everybody's trying to come up with ideas, but in the end, ideas need to come from everyone. So you can't be too precious about your own ideas. You need to take in insights from everyone. So people sort of leading teams and projects, there's a balance to be had, and you need to be trying to draw out insights from as many people as possible, everyone in the team, and pay attention to when people have ideas, and even if you initially disagree with them, to still try and understand them and learn from them. So
perseverance is key in science. And in your career, a time where like a molecule looked really exciting at first, but the team or like the project, just how it progressed, it realized it was not going to become a good drug or a good sort of candidate moving forward.
The project I worked on for about 10 years at one point failed in the clinic, and it was like, oh, it didn't fail in the clinic. It actually did what it wanted, but we turned up some entity that we hadn't seen before, and that was okay, right? We can't go any further with that. So that was very disappointing.
And how did you navigate that disappointment? Because it's hard when you spent so long working on something and it fails in a way that's so out of your control.
That one was actually so far into development by the time it failed that I was continuing to be involved in the project, but it wasn't actually my primary activity. So I was already engrossed in, I think, probably 2 other projects at the time. So yeah, it's kind of like I was already involved in other things. So yes, it's disappointing, very disappointing, but you're already engrossed in something else, so you move on. It can be really tough when projects that don't nominate a candidate, there's then a decision has to be made. Are we going to continue to resource this project, or is now the time to stop it? And there's never a clear cut. Sometimes you will run a key study, for example, a tox study, and you see that you've got undesired tox. And if that undesired tox is related to the mechanism that you're investigating, then your project is dead. And that's a clear-cut decision. But sometimes, so a particular project I'm thinking of, we worked and we overcame problems, and then we hit more problems and we tried to overcome those. And we were kind of stuck in this apparently never-ending loop of you solve one problem and then you hit the next one. And there comes a point where you have to say, are we ever going to reach a candidate? And that's a judgment call. It is not, and in that case, it was not clear-cut as to whether we stopped the project or carried on. And this then involves discussions with management and between management and the project team. And ultimately, we decided to stop that project. And you never know.
Yeah.
If we'd carried on for another 3 months, 6 months, would we have found something that was good enough? Or had we reached the point where, we tried so hard for so long, we were never going to get there with our current series. And that was the position we came to. But yeah, it was a judgment call.
And do you think that sort of ability to judge whether that sort of no-go decision or that yes or no, do you think that comes with experience or time, or is that something that you can learn?
I mean, you learn it, but a lot of it is through experience. I mean, it's one of the things that the senior managers who review projects, they're all very experienced. And being in senior management and reviewing the whole portfolio of a division, they get experience across a lot of projects. So when you're working at the coalface, as it were, of a project, you're really focused in on your project, whereas the management level, they're looking at all the projects in the portfolio, and I mean, essentially, they're making bets, and they're betting with the company's money, and they're making a decision as to which of those ones are worth going on with. If you're doing something, then by definition, you're not doing something else. So, I mean, there is a really important role, and it's very important that there's an experienced medicinal chemist, at least one experienced medicinal chemist in that management group, who is able to give the medicinal chemistry insight into whether or not projects should stop or proceed.
And I think that can be a hard thing to do. It requires having the experience and knowledge, because you're right. You could have spent 3 months more and it could have worked, but equally you could have spent 30 years more and it could not have worked. So switching track slightly, in 2011, you were writing and you were saying that a corporate reorganization led you to move from England to Sweden. So you've moved countries for your career, which is a big thing. How did that shape you professionally and personally?
I mean, it seemed like a really big move to come to Sweden. Actually, for me, it worked out extremely well. I wouldn't say I was stuck in a rut, but the opportunity for a fresh start in a new group was really beneficial for me. It took me out of my comfort zone, and I had to learn new ways of working, new colleagues, essentially completely new managers. So it was hard in that respect, but that really was actually led to a lot of growth, and it provided opportunities for me that I probably wouldn't have got previously. So in terms of my career, it was probably the best thing that happened to me in my career in retrospect, even though at the time it seemed really, really hard. Personally, I found coming to Sweden really, really beneficial. I've absolutely loved living in Sweden. To my surprise, having not enjoyed learning languages at school, found I really enjoyed learning Swedish, and I've really settled into the country and the community. I've become a citizen. So it's been a fantastic opportunity.
And what advice would you give to someone considering moving for a scientific role?
You have to like science. You have to like the process of going to the lab, making compounds, and then sitting at your computer, which is how you'll do it these days, and looking at data and thinking about the results, thinking about things. So if you enjoy that aspect, and not everyone does, then it's a fantastic career. And I can truthfully say that I have looked forward to going to work essentially every day of my career. And that's not true of many professionals in many careers. So I feel incredibly privileged to have had such an interesting job, and a job that I think overall is still very worthwhile to do. I think there are still a lot of diseases and conditions that are not very well treated. So yeah, it's been a huge privilege to be part of that.
And I mean, when you look back on your 37 years in the field, what would you say you're most proudest of?
I mean, the answer is projects to progress. I think I'm very proud of that. I think these last few years I had the privilege of being a project leader, and working with incredibly talented younger scientists, and realizing that from my experience, I was able not to do things, but to help them do their roles effectively for us as a team. I think that was really rewarding, that sense of, I mean, it was really fun being at the center of the project, and interacting. I mean, as a medicinal chemist, you interact a lot with other colleagues, but as the sort of project leader, you interact more with more people more consistently, and being able to help and guide younger colleagues to success and to progress was extremely rewarding.
Mentorship is so key, especially if you've spent so long in the field helping to bring the next generation of medicinal chemists. We're coming to the end of the episode, but is there any other advice you would give someone early in their career who's interested in medicinal chemistry or drug discovery?
If you want to become a medicinal chemist, I mean, it goes back to where I started, that as a medicinal chemist, you need to be able to make any molecule. So we require people who are very skilled synthetic chemists, and I wasn't the greatest synthetic chemist ever, but I've worked with some really talented people. But we do require people who can make molecules, but it's a much broader role than that. Yeah, it's about creativity. It's about collaboration. What advice? And there is a lot to be learnt. So you've never arrived. I mean, that's because science changes. Science constantly goes forward. You can't go into a role and just settle there. You need to constantly be learning new things, keeping abreast of the changes that are coming. Yeah.
And it'll be exciting to see where the field progresses. Right now, I'm sure there are things that you probably couldn't have imagined being developed that are developed. And it's going to be interesting where in the next 37 years, what's to come.
Yes. Yeah. And science, being part of science is fascinating.
I agree. So as we finish, I always like to remember that people are not just their work. Interesting as their work is. So what do you like to do outside of what you do professionally?
I mean, probably my biggest outside activity is that I'm a Christian. So I'm a member of a local church. For quite a few years here, I was actually a sort of leader on a church. I used to preach regularly. I've taken good advantage of the wonderful natural world in Sweden I have a windsurfer on the archipelago near Gothenburg. I've gone walking in the woods and the mountains, and I have a garden that I like growing things in. So those are my sort of, and my final interest is that I play board games, strategy board games.
Oh, well, Matthew, thank you so much. I really appreciate you taking the time to just reflect and share some really helpful advice on your career and your experience working in this field.
Thank you, Menashe. It's been a privilege to join you. I hope that was of interest to you and to all who listen.
Well, and thank you for listening to Meet the Medicine Makers. This podcast explores the people behind how medicines are made, from early research through to real-world impact. If you found this episode useful, feel free to share it with someone who might benefit, and you can find out more about Matthew and his work in the description. See you in the next episode.